Our next speaker is a wonderful translational investigator. Professor David Katz is Nello Teer Professor of Biomedical Engineering. He is also a professor of obstetrics and gynecology. As you've heard already today, he's been tremendously supportive to some of our clinical investigators. And today he's going to talk to us about application of engineering sciences in HIV prevention.
We have to have some engineering toys. Dave Walmer gave you a wonderful example of how low-tech with some imaging and engineering can lead to high impact. And we do a lot of things in engineering, and I'm going to share with you some of those today. And we do a lot, some of the recent things. And in fact, at Duke, we do a lot of engineering related to HIV. Stefan Zauscher in the Pratt School works with Munir Alam on creating new materials that simulate the viral envelope. This is material science, you might say. We build a lot of imaging devices in our group, our extended group, going back to our early years with Dave Walmer, and recently we ask a lot of questions with devices that are inserted vaginally about the integrity of the tissue, what's on it, are there drugs there? And I actually thought about giving this talk about intravaginal imaging today, but I thought I'd give you an example of a different side of engineering. It might be a little more Big Bang Theory relevant, but it's no less valuable. And this has to do with the use of computation in design, as well as in understanding biology and its translation into medicine. This work, by the way, involves partnership with the CFAR UNC, the Angelica Shuba. And finally, we actually work with behavioral scientists at Brown, Kate Morrow, and to some extent Ken Mayer, looking at how sensory perceptions and preferences about things that people in their bodies actually relate to the properties of materials. And there's engineering in this. It's a little bit like what they do in food science. But today I'm going to talk about something different. I'm going to talk about transport, how things move. And there's a lot of this involved in the natural history of HIV and how we try to mitigate it, either from a prevention point of view or from a therapy point of view. And I'm going to be talking primarily about drugs that we put in the body to try to prevent infection. But a lot of the stuff that I'm going to talk about applies to how antibodies work, certain biophysical aspects of that, maybe not quite so conspicuous, but it does matter. And again, I'm going to focus on the prevention side. So I've got just a cartoon here of events that you're all too familiar with. As we have a cartoon of the vaginal mucosa, and we watch cell and cell-associated and free virus migrate through the epithelium to host cells, where they bind and begin to replicate. Now, there are different places that we can target interrupting this process, having to do with the viral envelope, having to do with entry of target cells, having to do with viral replication. And in fact, the drugs that we study to deliver to try to interrupt transmission address all of these. Entry, or the viral envelope, entry, replication. Now, we know that oral PrEP with ARVs works, but not everybody will use oral PrEP. And the truth is, from a pharmacological point of view, although it works epidemiologically, pharmacologically, it's okay. But behaviorally, as well as pharmacologically, we need alternative methods that either are of an on-demand type or something that's long-acting. And that's where I'm going to talk to you today about one of the places where we as engineers hang out. Now, microbicides have been, the term microbicides was coined not too far into the AIDS epidemic. And these come in different forms. They're different types of drugs, and we put them in the body using different delivery systems. We use gels, we use intravaginal rings, sort of mimicking NuvaRing, and I'll say a little bit about that at the end of my talk. We use films, we use tablets, we use suppositories, we use implants, we use injections, all of the above. And each has its strengths and its weaknesses from a pharmacological as well as from a behavioral point of view. Now, gels were one of the early dosage forms that was applied to try to introduce drugs that would be effective against the virus intravaginally. And as most of you know, there were 3 big phase 3 trials of a gel containing tenofovir, which we know about for therapeutic uses. And the first trial worked, Caprissa. The second trial, Voice, and the third trial did not work. Why? Well, poor adherence in the Voice and the FACS trials has really been implicated as the culprit. And in fact, recent statistical analysis of the Voice data shows that in that subset of users who did apply the gel reasonably well, it did work. I'm using very simple language to describe a lot of epidemiologic and statistical detail. The story is if you use the gel, it worked. And in Caprissa, if you use the gel more, it worked better. So gels will work, but we've got to make gels that people will be willing to use. And the gel that was used in this trial was not designed by a process that we as engineers would call anything remotely like rational design. So we're putting our nerd hats on here and saying, so how do you design something for this X-rated part of the body? Can we develop a microbicide product that works and that people will be willing to use? And there are a lot of ways we can look at this. What about the drugs, the cocktails of drugs? If we're talking about a gel, I'm going to use a gel to illustrate the story, because the truth is, although gels are probably not going to be the last word, to say it, put it mildly, we know a lot more about them than any of these other dosage forms. And so we have more data. How do we apply the gel? What volume? How frequently? And of course, we have to educate people in proper gel use. But these are, with the exception of the educational component, even of that to some extent, these are areas where we can attack with our engineering toolbox. So, let's get at it. The vaginal epithelium varies during the menstrual cycle. It challenges us because there are very few mammals that have an epithelium in the vagina like us. Pigtail macaque is just about the only one that's got it, and the good thing about a pigtail is it's got a menstrual cycle. So it's a monthly cycler. Even rhesus is a seasonal breeder. So here's a cartoon of what it looks like. And again, it's a race of the virus and the drug. Now, I'm going to talk to you about the way we study how this works more than how we measure it. We do spend a lot of time trying to figure out how to measure it, whether it's where the drugs go or where the virus goes, or how they bind to each other, interact, and other people here do this as well. So I'm going to talk about something that's a little different, that's very dear to our heart, and that is you can use mathematics in modeling to explain cause and effect. In this case, what we call mass transport. Where does the drug go? Where does the virus go? So we make models of drug delivery. They're different from the traditional PK models because they're deterministic models. And they predict where the drug is. What's its concentration in the epithelium, in the stroma, in the blood, in the vaginal fluid, in the lumen, as functions of time and space. And to build these, to successfully use these models, however, we need data. And so we have to, in this particular context, we have ended up having to create some new experimental methods to get the data that we need for these models. And I'll tell you a little bit about that. Now, here's an example of an early model that we built. David Montefiore helped with this, with a grad student, Anthony Giannotti. And this was for a protein from NCI called cyanovirin, which it attacks the proteins on the viral envelope, and it's no longer considered a really attractive candidate as a microbicide, but there's a new protein called grafithsin that is. And so the lessons learned from this analysis apply to that. You can make a mathematical model, a deterministic mathematical model of the co-transport of virus and semen, and let's say cyanovarin, or by extension grafithsin. Let's say in this case we've got a gel layer. And you can make a mathematical model of how they mingle. So what I've shown here, and there's a lot of math that goes into this, but there are no equations today. But what the point is that the model tracks where the drug goes and where the viruses go, where the virions go, when they collide, how frequently they collide, how they stick, what happens when they stick, how many molecules you need at any one time to keep the virus neutralized. You can take that information, and here's the kind of stuff that we love. These are called heat maps. So the color is whether the virus is infectious or not. Time is on the x-axis, and the y-axis, the upper half is the semen layer, the lower half is the gel layer. You can look at these colors, and with the exception of the upper right-hand corner, that's the drug. That's where the drug goes. But in this case, because we know a fair amount about how cyanovarin interacts with the virus, we can explore where the infectious virus is and whether the drug is neutralizing it before it gets through the gel and down to the tissue. So red is bad, blue is good. So we can ask questions like, if you have the gel layer, don't have as much gel there, how much of a difference does it make? Suppose the drug isn't as potent. So my purpose here is not to go into the gory details of this particular analysis, but just to tell you this is very rich information from a biological understanding point of view, and from the translation of that understanding into our conception and perceptions of better products. Now, the tenofovir gel has had a lot of study. Again, we don't think it's a particularly good gel, and really this work is motivated by the question, can we use less gel and still be just as effective pharmacologically and have the gel more attractive to users? And the answer is yes, we think you can. And here's some of the evidence that we are creating in support of that. We have created mathematical mass transport models of the transport of tenofovir from a gel into the vaginal epithelium and stroma and thence into the bloodstream. To do this, it's kind of complicated because when you put a gel in the body, it's got to spread. And and the drug is diffusing while the gel is spreading, so we divided and conquered. We looked at spreading and we looked at drug diffusion separately, and then we put them together. In so doing, we had to measure drug concentrations to get some of the parameters that go into the model, the so-called diffusion coefficients, and I'll tell you a little bit about that. Yeah, my problem is I can't see my talk on the screen here, so I have to turn around. Oh, maybe I'll go around like this. So the yellow are the key points here. Study gel spreading, study drug diffusion, put them together. Build some instruments to measure drug concentrations and take those data and compute the parameters that go in the models, then use the models to ask meaningful questions. And these are not your typical PK models. Here's an example of some results by Jing Gao, one of our students who's sitting here in the audience, that shows something that the models predict. This is the concentration distribution profiles of tenofovir as a function of time after it starts to diffuse in the gel through the vaginal epithelium and stroma and into the blood. And you notice how steep these profiles are. The goal is, are we getting enough drug where it needs to go to neutralize the virus, and are we getting it there before the virus gets there? That's the bottom line, right? It's a race. And this is the kind of raw information that one uses to ask that question objectively and to translate what you learn into product design. Now, We took the predictions of this model and we compared it with some PK data, which are in parentheses. So these are some of the standard pharmacokinetic parameters that you compute when you look at drug delivery, drug distributions. And if you see this, you know, the numbers in parentheses and the numbers that we predicted are pretty close. We were amazed. The area under the curve numbers are a little different, but the way they calculate area under the curve in PK in standard pharmacology is a little approximate, shall we say. So we weren't surprised that they were not quite as close there, but we were very pleased with this. Now, we needed parameters in the models. So we had to get these parameters. How does the drug move? In traditional pharmacology, you do it's called a permeability assay, and it doesn't tell you a whole lot about how fast the drug goes, where it goes, especially when you have tissue with two very distinct layers, an epithelium and a lamina propria. And so we needed to measure this. So Palm Chushen, who's another grad student in our lab, said, okay, let's use Raman spectroscopy as an optical technique. We know a lot about light in engineering at Duke, and in biomedical engineering in particular, so-called biophotonics. We build a lot of devices. Dave mentioned this in his talk, and he mentioned Nimi Ramanujam's work using optical probing techniques to look at the cervix. We use an alternative optical technique to look at the cervix, and light works. So Raman spectroscopy is a technique that is very specific to the molecular details of targets. Its challenge is that it's not super sensitive. But we created an assay using confocal Raman spectroscopy to track drugs that migrates through tissue. And this is just a cartoon of this. You can get these concentration profiles. Oops. So you can get concentration profiles. And we built this is Palm's work. So we created a Transwell assay where you harvest some fresh, in this case, porcine vaginal tissue. You keep it happy. I'm sparing you a lot of the details. And you do Z-scans as the drug diffuses through the tissue. And you process those data, and you can get diffusion and partition coefficients of the drug. This is the holy grail of transport. These are the parameters that we teach when we teach mass transfer to engineering students, and they go into the models. And they then enable us to make more detailed, more accurate predictions about the natural history of where the drug goes. We were originally using commercial Raman instruments, and they had some disadvantages, so we started building our own. And this is the bench in Adam Wax's lab in biomedical engineering, so we build our own devices now. And like I said, we build a lot of stuff that uses light to look at tissue and to look at drugs. This is an example of one of the early ones, and we get higher fidelity measurements when we do it. Now, back to the gel. The gel spreads, it may leak. We need to understand the mechanics of how that works and then combine that with the drug diffusion. And Jing Gao figured out how to do that. And so this is just a cartoon on the left of different locations where a bolus of gel might be in the vagina. Notice that in one of them it's leaking. We can keep track of leakage in a model. We can compare it with what we measure. And I should say that complementing this, and I guess perhaps I should have mentioned it, we do imaging studies where we look at gel distribution in the vagina and we ask questions about, are there differences between gels with different properties and different volumes? And the answer is yes. And what the modeling tells us is, do we care? And the answer to that is yes. So here's an example of one of these heat maps for tenofovir, but we've also in this model, we've got, just to remind you, tenofovir works as a reverse transcriptase inhibitor after 2 phosphorylation steps in host cells. And so it gets converted to tenofovir diphosphate. So when you build your model, you have to work into it the kinetics of the conversion of tenofovir to tenofovir biphosphate. So it's a little more mathematical work, some more terms in the equations, a few more equations. That's what we do for a living. And so here's some heat maps that illustrate the gory details of this. So on the left is tenofovir, on the right is tenofovir diphosphate. The gel is spreading from right to left, and it's spreading downwards into the tissue. You can kind of see the leading edge of the gel. And these models tell us how fast it takes the gel to spread and what the drug concentration profiles look like. And then we can convert these data to the kind of numbers that pharmacologists need to start to think about the effect of PK. So these are plots that are tried and true to any PK study, where you're taking the average concentration in the epithelium and the stroma and in the blood, and computing how that varies with time after you insert a gel. And in this case, we've looked at several different gels with different properties. We've looked at different gel volumes. Oh, I can design gels with different properties. I can put them in at different volumes. Does it make a difference? The answer is yes, it does. But do we care about the difference in drug delivery? I mean, it's all well and good to do, this is PK, but really what we need to know is PD. Is it working against the virus? So we're starting now to do this. We have some idea of the concentrations of tenofovir and tenofovir diphosphate that are effective to neutralize virus. These data are imperfect, and they give us great pain because of the way they're collected, because there's some logical gaps in the way these EC50s and EC90s are calculated, but they are a reference, so what do? We look at upper and lower bounds of them, and we do the following. Oh, this is tenofovir diphosphate, one more plot. This is why tenofovir works so well. These are the concentrations of tenofovir diphosphate in the stroma, where a lot of host cells are in the vaginal mucosa. And we've looked at different gels, we've looked at the site of placement of the gels, but the point is these curves flatten out with time. Tenofovir diphosphate has a long half-life. That's why, it's one of the reasons why the drug is so good. It stays in the target cells for days, which is contrasts it from most of the other antiretroviral drugs. That's good. That's very good. Now, do these levels of tenofovir that we deliver to the stroma work? Now, this is a new concept. Basically what we do is you say, okay, we know how much drug is there. We have a target value of the drug. So let's look at what fraction of the target tissue has got a concentration that equals or exceeds the target value. A simple concept. We call it percent protected. What fraction of this, and by direct inference, what fraction of the target cells in the stroma have drug concentration in them as a function of time that's greater than equal to some target EC50 value? So you get plots that look like this. This number goes up and then it levels off. Now what we've done here is we've looked at 3 different gels with different properties, and we've looked at a range of values of the EC50. This is what it looks like. And you can see that the curves level off at different levels. The initial kinetics is rather similar, but it tells you how long you have to wait, basically, after you apply the gel before you've got prophylactic levels of drug where you want it to go. That's what you need. That's the translation of PK to PD. And that's the kind of thing that engineering can contribute. Now, just a little bit, we're not wedded to gels. We've done some work. Jing is leading this on intravaginal rings. And also we're looking at the rectum as another compartment because there's a lot of interest in delivering ARVs rectally because there's a lot of rectal transmission. I'll say a little bit about that. Just quickly, you can build mass transport models. There are a number of intravaginal rings being developed right now. Some have ARVs. Many of them have ARVs and levonorgestrel, and then we're another progestin, the idea being that you're producing a product that's both contraceptive and prophylactic against HIV. Our concerns about progestin and HIV notwithstanding, and they are considerable, it's a work in progress. You can build these models for these intravaginal rings, and here's just one plot that illustrates their results. I've compared on the left. Let's see, we've got a gel on the left and a ring on the right. So this is that tenofovir gel that I showed you recently that's in the Conrad, was in the 3 phase 3 trials. And what I want you to focus on is focus on the blue line. The blue line is tenofovir diphosphate. How quickly does it rise before it levels off in a gel versus a ring? There's the ring. There's the gel. You get more in more quickly with the gel, but of course the gel leaks out eventually. But these are the kind of numbers that help us to, ask questions like, well, how long do I have to wait before I insert a gel? How long do I have to wait before I insert a ring? If I take the ring out, what happens? These models give us ideas about that, and I'll wrap it up in just a minute with how we use that. We've also looked at the rectal delivery of drugs. The rectal mucosa is very different. It's got crypts in it. And the epithelium is a single layer of columnar cells. It's quite different from the stratified squamous epithelium in the vagina, and it doesn't vary during the menstrual cycle. The stratified squamous epithelium in the vagina does. But you can build models. Now here's an oversimplification of a real nerdy view, the way an engineer looks at these crypts in the rectum. You say, okay, we've got these tubes, and we can get dimensions for these things, and we've got a canal in the middle. Now this is, what we create is more sophisticated than this, but this is the concept. You don't have it's kind of a relatively flat surface now. You have these little crypts, and you've got to work out all the mass transport for how the drug goes in there. Well, Jing has figured out how to do that, and so here's the model. I think I'm one slide out of phase with you when I'm talking. I'm sorry about that. Here's an example for a heat map in a single crypt. And you can see how the drug diffuses. Now this is the case, this is for an enema. We're working on a tenofovir enema project with folks at Johns Hopkins. And this is 6 minutes after you've inserted it. Now, final slide here. This is a PK plot for that model for crypt, and we conceptualized the mucosa in the rectum as an ensemble or a mosaic of crypts. So you're basically adding up what happens in these crypts to look at the total drug. What I think is interesting here and quite relevant is how quickly drug gets into the rectal mucosa versus the vaginal mucosa. It's hardly surprising because the epithelium is so much thinner in the rectum, whereas in the vagina, it's what we call the rate-limiting barrier to getting a drug to the host cells in the stroma. So the vaginal gel data are shown in purple here down at the bottom. And all these others are questions. These are variations in the parameters of the application of the rectal enema. Big difference. And these different colored curves are curves that are based upon questions that we're asking about how long do you retain the enema, for example, before you expel it. And so this is at the beginning, but the point is we can ask some very meaningful questions using modeling to complement all the experimental work we do both on the bench and in people and in animals, to ask very, very important questions about how infection occurs and how we can design products that can help to prevent that. As we like to say, we use modeling to explain cause and effect. And you know what? It works. It works in the nicotine patch. There are a lot of areas in drug delivery in the body where modeling really helps us understand the biology and the pathology and the pharmacology of the system, and then design products to achieve targets. Unfortunately, the HIV/AIDS field hasn't used this so much. There's some very elegant modeling of the spread of the virus itself from a more of an epidemiologic point of view. Alan Perelson, who some of you know, does really beautiful work. That's not down to the nuts and bolts of where the drug goes and where the virus goes, and that's what we're trying to do. And again, the concept is with this information, we can rationally design products. We can simulate a lot of variability in the design parameters, in the host, the size of the vagina, the variations in the amount of vaginal fluid, the thickness of the epithelium, all the stuff that varies during the menstrual cycle. This comes out of the wash in the modeling, so we can replace an awful lot of experiments early in the understanding and the design process before we go to experiments. And so that's one example of what we do in engineering, and it complements all this other elegant experimental work where we in the Pratt School at Duke want to work with you guys to try to figure out lots of other binding sites in applying our craft to help us all understand HIV and AIDS. I'd just like to thank some of our collaborators here. We've got a couple of students from the lab, Jing Gao and Morgan Simon, sitting in the back. And as I said, we work with the CFAR at UNC. We work with the CFAR at Brown. And we work with a lot of people. These are kind of the core players in the most recent work that I'm showing you. And when we started the imaging, Dave Ulmer was here at Duke, and our first imaging studies were together with him, where we published a bunch of papers together. And finally, I just want to say we teach this stuff. I teach a course on HIV for engineers. I'm teaching it right now. And this stuff also comes up in some of the other transport courses that we teach. And one of the things I'll just conclude was we've got this new T32 that sort of is in the spirit of this. We thank Georgia, we thank Guido for all their hard work in putting that together. We're going to try to figure out how to combine this stuff more effectively in the way we train our more advanced students and postdocs in the future. So engineering matters. We've had CFAR support in the past. We very much appreciate it. We've got some new ideas that might be of interest to the CFAR. And like I said, we want to work with all of you. Thank you.